Good afternoon, everybody. My name is Leah Trieu. I'm a first year student at Riverside City College, and today I'll be presenting on the antibacterial activity of natural versus modern antibiotics against the OP50 strain of Escherichia coli. The two main purposes of my research was to initially examine the effectiveness of natural antibiotics against the OP50 strain of Escherichia coli using the Kirby-Bauer diffusion method, and secondly, to compare the susceptibility of E. coli OP50 to natural versus modern antibiotics. The reasons I chose to focus my research on antibiotic resistance and specifically natural antibiotics is because initially the CDC regards antibiotic resistance to be one of the most significant public health challenges in the modern day. Bacteria rapidly evolves and changes, and quite frankly, science cannot seem to keep up with all of these evolutions. In the US alone, at least 2.8 million people each year will receive an antibiotic resistant infection. This is a very concerning statistic. And secondly, as someone who hopes to serve as an infectious disease physician working in rural and underserved communities, I'm a firm believer that healthcare should be a basic human right. And in areas where civilians lack access to any medical centers, hospitals, or when patients simply cannot afford to seek the care they need, I became extremely interested about naturally derived antibiotics and how they're employed in place of what we consider to be traditional modern medicine. Lastly, as someone who has spent hundreds of hours volunteering in ICUs and surgical units, I have noticed that E. coli infections were a common occurrence among long-term patients or patients who have received several different medical treatments. The first crucial piece of literature to beginning my research was Antibiotic Resistance and Extended Spectrum Beta-lactamases, Types, Epidemiology, and Treatment. So what exactly makes E. coli such a difficult bacteria to treat? First, E. coli contains porin proteins in the outer membrane, which allow it to have a selectively permeable cell membrane that gives the opportunity to bacteria to block out the entrance of antibiotics. Because they have double membranes, this makes it increasingly more difficult to kill E. coli. And E. coli can also produce beta-lactamase enzymes, which will break down the structure of antibiotics and leave them completely ineffective. Now, discussing my investigation into natural antibiotics, in the study, honey, its medicinal property and antibacterial activity, has been shown that Manuka honey is effective against various strains of E. coli and other antibiotic resistant bacterial strains due to the hydrogen peroxide in Manuka honey. This creates a very acidic and toxic environment for bacteria to survive with low pH levels and high osmolarity. The next study that was crucial to my research was on tea tree oil, a review of antimicrobial and other medicinal properties. Tea tree oil has been used for centuries as an antibacterial, antifungal, and antiviral treatment. There has been no clinical antibiotic resistance ever reported on the use of tea tree oil, which made it extremely interesting to me to include in my study. And tea tree oil has been shown to have detrimental effects on the structural and functional integrity of these bacterial membranes. Next is the study antibacterial and antioxidant activities of mentha piperita L. So in peppermint oil, there are a number of phenolic compounds and flavonoids that have been shown to damage bacterial membranes and inhibit the growth of bacteria. Furthermore, peppermint oil contains high concentrations of menthol, which has demonstrated antibacterial effects against both gram-positive and gram-negative bacteria. Lastly, the study effect of essential oils on pathogenic bacteria investigates oregano oil. Oregano oil also contains phenolic compounds, carvacrol and thymol, which have been shown to disintegrate the outer membrane of gram-negative bacteria and completely disrupt regulatory functions, which then kill the bacteria. Based on the literature and the studies that I have reviewed, what I learned led me to the hypothesis that my chosen natural antibiotics would be more effective against this strain of E. coli and produce larger zones of inhibition than the modern antibiotics I would be testing. These modern antibiotics include penicillin, erythromycin, ampicillin, and neomycin. Out of these four, erythromycin is not a good treatment for E. coli infections because of the selectively permeable double membrane that erythromycin just simply cannot surpass. And penicillin, although a very common treatment for other bacterial infections, does not work the best against E. coli because E. coli's ability to produce beta-lactamase enzymes, which then break down such β-lactam antibiotics. For further clarification, the Kirby-Bauer diffusion method is used to test antibacterial susceptibility, and a zone of inhibition is the circular area around the spot of the antibiotic where the bacteria colonies do not grow. For my methodology, the first step I conducted was preparing the E. coli suspension by diluting Luria-Bertani broth that had already been colonized with this strain of E. coli and diluting it with phosphate buffered saline to obtain a McFarland turbidity standard of 0.5 in order to follow standard procedures for the Kirby-Bauer method. Then I measured with a pipette and spread 0.1 milliliters of the suspension onto 11 different petri dishes containing Luria-Bertani agar. Next, I took blank antibiotic discs and impregnated 20 of them with the natural antibiotics using a pipette. 5 of each for the different natural antibiotics. And for the modern antibiotic discs, I had ordered them online, so I didn't have to make or prepare those myself. Now, getting into the Kirby-Bauer methods procedure specifically, initially on the bottom of each petri dish, I used a Sharpie just to divide the petri dish into 4 equal sections to serve as quadrants for my disc placement. And I also labeled each quadrant with the names of which antibiotic disc would be placed where. On place 1 through 5, I placed one of each modern antibiotic disc in each quadrant and did the same using the natural antibiotic disc for plates 6 through 10. I left the 11th dish empty in order to serve as a control dish. And on each plate, I also left a completely blank antibiotic disc as a further control variable. Then after all of the discs were placed onto my petri dishes, I placed them into an incubator for 12 hours to allow the bacteria to grow. And I chose to incubate them at 37 degrees Celsius in order to stimulate actual human body temperature. After 12 hours, I removed the plates to measure and record the zones of inhibition that had been produced thus far, and then placed them back to incubate for another 12 hours. After the total 24 hours had surpassed, I removed the plates again to measure and re-record the final zones of inhibition produced. So now moving on to the results, this table depicts the zones of inhibition produced by the modern antibiotics after 12 hours of incubation. So as we can see here, the erythromycin produced no zone of inhibition at all, which was to be expected as it cannot penetrate the selectively permeable membrane of the E. coli. And penicillin produced the second smallest zones of inhibition, which was also expected due to E. coli's ability to produce these beta-lactamase enzymes. At this point, we can notice that ampicillin and neomycin produced relatively similar results. They're very close to one another. And now moving on to the results for the natural antibiotics, the zones of inhibition produced after 12 hours, we can see that Manuka honey actually did not produce any zone of inhibition at all on any of the 5 plates. And the most shocking discovery to me thus far was that oregano oil, after 12 hours, had produced the largest zone of inhibition out of all the antibiotics tested. On this table, we can see the zones of inhibition produced by the modern antibiotics after 24 hours total. Erythromycin still did not produce any zone of inhibition, and penicillin zone of inhibition remained the second lowest of the 4. But before, when we saw that the zones of inhibition produced by ampicillin and neomycin were relatively the same after 12 hours, we can now notice that ampicillin had a greater effect on the E. coli than the neomycin did, and produced the largest zone out of all of the modern antibiotics. After 24 hours for the natural antibiotics, oregano oil remains the most effective antibiotic tested and produced the largest zones of inhibition out of all of them, surpassing ampicillin for every dish by at least 4 millimeters. After 24 hours, we can also notice that Manuka honey produced an extremely small zone of inhibition of 1 millimeter on only 2 out of the 5 petri dishes, and peppermint oil remained the second least effective natural antibiotic after both 24 and 12 hours. This chart simply displays the average zone of inhibition produced by each antibiotic tested after 12 and 24 hours. Erythromycin and Manuka honey were the least effective antibiotics tested, while oregano oil was clearly the most effective. We can now see how after 12 hours, the zones of inhibition for ampicillin greatly surpassed that of neomycin, despite being nearly the same at 12 hours. And even just observing the standard error bars for ampicillin and oregano oil would have still produced a greater effect than ampicillin at its lowest value for the margin of error. So connecting back to my original hypothesis, it was both correct and incorrect because although oregano oil, a natural antibiotic, produced the largest zone of inhibition compared to the modern antibiotics, on average, the modern antibiotics produced a zone of inhibition that was 4.9 millimeters greater than the average zone of inhibition produced by the natural antibiotics. The most surprising discovery I made after conducting my research was how effective oregano oil was at killing E. coli and inhibiting growth, which can definitely be attributed to its combination of phenolic compounds and its ability to completely compromise the structural and functional integrity of the bacterial membranes. I also found it quite surprising that Manuka honey barely produced any zones of inhibition. I theorized that this may be because I simply did not place enough of it on the antibiotic discs, or because maybe there was already bacteria present in the honey bottle and it was not sterile before application. Furthermore, if I were to continue and extend my research, I would replace erythromycin with another antibiotic, such as azithromycin, as the results of the erythromycin were to be expected. And I would also use a Mueller-Hinton agar in the petri dishes instead of Luria-Bertani agar in order to strictly adhere to the standardized procedures that the Kirby-Bauer method lays out. I would also like to expand the variety of antibiotics tested to at least 20 total, 10 modern and 10 natural. And I'd also be very interested in replacing the antibiotic discs over time with freshly impregnated discs, but placing them on the same petri dishes in order to see if I could provoke some sort of antibiotic resistant reaction from the E. coli. Lastly, I would like to thank my research advisor, Gina Brotman-Maduro at the UCR Molecular Cell and Systems Biology Department for allowing me to use her laboratory and guiding me in conducting my study. And that includes my research presentation on the antibacterial activity of natural versus modern antibiotics against the OP50 strain of Escherichia coli. Thank you for watching.